Hoje, no Entrevistando Cientistas, entrevistaremos o pesquisador, professor e oncologista Anthony Letai. O professor Letai recebeu da Universidade de Chicago seu título de MD e de PhD. Posteriormente, ele completou sua residência médica no Hospital Brigham and Women's e se especializou em hematologia e oncologia no Instituto de Câncer Dana-Farber. Em 2004, o Dr. Letai tornou-se pesquisador independente no Instituto de Câncer Dana-Farber e na Faculdade de Medicina de Harvard, onde hoje atua como professor de medicina. Seu laboratório estuda a evasão da apoptose, especialmente por células cancerígenas, e como essa evasão pode ser detectada e direcionada. Assim, nós gostaríamos de perguntar, primeiramente, como surgiu o interesse em estudar a associação entre apoptose e câncer.
Partly really just by accident. About 20 years ago, more than 20 years ago, I was a trainee and I wanted to find a laboratory to work in that would be where I could learn more about how to do research. And I really didn't know anything about apoptosis or programmed cell death, but I knew that the man running this lab, Stanley Korsmeyer, was very well respected, was a nice guy, everyone loved him. So that was the main reason I joined the laboratory, and that was the main reason I got involved in apoptosis. It was really by accident. I really chose the field more because of the person than because of the specific area. Of course, I subsequently came to understand it better, but I understood very little when I started.
A pesquisa do seu laboratório se concentra em como as células decidem se comprometer com a apoptose. Por que isso é importante para o câncer?
Partly because we wish all cancer cells would undergo apoptosis, right? And most effective therapies work by forcing a cancer cell to undergo apoptosis. So there's a lot of drugs that do that. Probably most of what we call conventional chemotherapy agents, probably most of them operate by making a cancer cell undergo apoptosis. But we've been studying probably most of the effective immuno-oncology therapies, like checkpoint blockade or even CAR T cells. They ultimately involve making the cancer cell undergo apoptosis. A lot of what I've worked on is just trying to make that work better so that we can, with a given drug treatment, make even more cancer cells undergo apoptosis.
Uma das técnicas estudadas pelo seu grupo é o perfil BH3. Você poderia explicar em que consiste essa técnica, sua importância na avaliação da dependência de uma célula cancerígena das proteínas antiapoptóticas da família BCL2 e qual sua relevância para entender os mecanismos de sobrevivência celular das células cancerígenas?
When a cell undergoes apoptosis, you can think of it as very much like a switch, or we like to think of it as like falling off the edge of a cliff. It's very much like a binary decision. The cell's alive and then it's dead. But you can imagine a cell might be really close to the edge of the cliff or a cell might be really far from the edge of the cliff. One of the first things we were interested in is there a way to figure out whether a cell is really close to the edge of the cliff or really far from the edge of the cliff? Because we thought that might let us understand why some cells are more sensitive to chemotherapy than others. The mitochondria and the permeabilization of the mitochondria is the step of commitment to apoptosis. What we did is we invented this BH3 profiling, which involves taking little peptides and sort of tickling the mitochondria and determining how much of the peptide you need to put on before the mitochondria permeabilizes. If you need to put a lot of peptide on the mitochondria, then that means it's really far from the cliff edge. If you need to only put a little on the mitochondria to see it first, then that means it's right near the edge of the cliff. And that allowed us to categorize patients' tumors as being very primed for apoptosis or very unprimed. Another way to use this, if you use a different kind of peptide, there's some peptides that only interact with, say, MCL1, which is one of the anti-apoptotic proteins. If it's very sensitive to that peptide, if you have a sample or a mitochondrion that's very sensitive to that peptide, that means it's an MCL1-dependent mitochondrion. There's another peptide that can probe BCL-XL and another that can probe BCL2. So in this way, we can figure out, even before there were drugs, figured out a way to categorize tumors as either BCL2 dependent or MCL1 dependent or BCL-XL dependent. And that matters much more now because now there are drugs and we can use this tool to identify where these drugs could best be used.
Qual é a diferença funcional entre linfócitos T e células CAR-T? E o que explica seu potencial de levar à morte células cancerígenas?
One of the most important differences between a CAR T cell and just a run-of-the-mill T cell is CAR T cells have been forced to express the CAR part. CAR stands for chimeric antigen receptor. And what it is you take the variable part of an antibody, those are made by B cells, and you kind of fuse it to a T cell receptor, and then you make that into a construct, and then you transfect the T cell. So now it expresses a CAR, a chimeric antigen receptor, so that that T cell gets activated when that CAR gets bound. Now, if you're smart about that antibody fragment that you choose an antibody fragment that specifically recognizes a protein that's on a cancer cell, but not on a normal cell. And that gets the cancer cells to specifically attack the cancer, CAR T cells to specifically attack the cancer cell. The first success in this was when people used CD19. Now, CD19 isn't perfect. It's present on most cases of B-ALL and a lot of different types of lymphoma. It's also present on normal B cells. So if you give someone a CAR T cell, you may well cure their acute lymphoblastic leukemia. You'll also wipe out their normal B cells, which are part of our immune system. But we can survive without our B cells. You'd rather have them around, but we can survive without them. And if it's a choice between having your leukemia cured and losing all your B cells or dying of leukemia, the choice is obvious. You should have your leukemia cured and have your B cells eliminated. So now what people are doing is they're trying to identify other antigens beyond CD19. A key part of this, linking it to apoptosis, many, if not most cells, when they're killed by a CAR T cell, it's not by magic. People often view sort of a magical event occurring when a T cell recognizes a cancer cell or a target cell and the cell explodes. That's not what happens. What happens is that CAR T cell releases factors that get into the target cell and activate the programmed cell death mechanisms of the target cell. So what we've been working on is sort of turning up, activating the programmed cell death mechanisms of the target cells so that when they are identified by a CAR T cell, it's easier for the cancer cell to die.
Como você explicaria a diferença de citotoxicidade das células CAR-T em diferentes tipos de tumores, obtendo, de acordo com o contexto experimental apresentado no estudo sobre a quantificação dos requisitos de apoptose mitocondrial na morte de células cancerígenas por células CAR-T, maior eficácia em tumores hematológicos do que em tumores sólidos?
Very good question. And really, I don't know the answer that clearly because really no one, there's a lot I don't know, but really no one really understands perfectly why it works better in some cases than others. But I think at the end of your question, you hit on an important part of it. I think because CAR T cells operate, one of the main ways they operate is by activating apoptosis. You know how I talked about some cells being more primed close to the cliff edge than others? You might expect that CAR T cells are going to work more effectively in cells that are closer to the edge. Now, we have done a lot of work on a lot of different kinds of cancer cells, and ALL, acute lymphoblastic leukemia cells, are a type of cancer that are very, very close to the edge. So to me, it's not surprising that they're most effective in the case of ALL. And I think one of the challenges in moving it to solid tumors tend to live much further from the cliff edge. So it's just going to take more to treat them. And that's something that we're working on here right now.
De seus trabalhos teve como objetivo elucidar a seleção de alvos terapêuticos específicos no tratamento deste câncer, especificamente miméticos de BH3, como venetoclax e navitoclax, que inibem BCL-X. Nesse contexto, qual a relevância dessas elucidações na seleção de terapias miméticas de BH3 para pacientes adultos com leucemia trazidas pela sua pesquisa?
So far we use very regularly, that is our FDA approved venetoclax for use in CLL and AML. But I think the time will come when virtually every disease will be using one of the BH3 mimetics. We may need to identify on a person-to-person basis which one is the right one for that person, because one drug might inhibit BCL2, another might inhibit BCLXL, another might inhibit MCL1. We don't immediately know which is the right one. I hope that we can use BH3 profiling to do some of that. We're a little spoiled, like in the case of CLL or ALL or AML, those diseases are pretty homogeneous. And the vast majority of cases are BCL2 dependent. I don't think we're going to find that in most other diseases. Like multiple myeloma, for instance, we know that maybe only about 20% are BCL2 dependent, and the others are MCL1 or BCL-XL dependent or some combination. And we're going to need a tool to identify which patient is which type so we can give them the right drug. I foresee, and I hope we get to the point where we can use BH3 profiling to do that.
Então, para finalizar, o seu grupo de pesquisa contribuiu significativamente para a aprovação pela FDA do tratamento combinado de venetoclax para pacientes adultos com leucemia mieloide aguda. Você pode nos contar mais sobre essa experiência de ver essa linha de pesquisa do seu grupo se concretizando na prática clínica?
Happy to, because that's one of the most gratifying things that I've been able to do. It's funny, though, I didn't view myself that I was going to be the person to do it. Early on, we were working in the laboratory, and it became very clear to me that we'd identified that AML is largely BCL2 dependent, so a drug like venetoclax would be good for it. And I thought just sort of like giving lectures and writing a paper about this information, that would be enough, and somebody else would take over from there. Well, that somebody else never appeared. And that's when I realized that everyone's very busy and not everyone reads everything you do. And sometimes if you really believe in something, you have to do it yourself. At the same time, I identified, I didn't know her before then, but she became a collaborator and a friend, Dr. Marina Konopleva, who at that point was at MD Anderson. She was using different methods, but she had determined the exact same thing we had. And so we decided rather than compete, we pooled our data, we put it together. And then I brought it, I traveled from where I live to Chicago where AbbVie is, they make the drug, and I presented mine and Marina's data to say, you guys really need to start a program. We really think it's going to work in AML. And then they decided on the basis of that to allow us to fund a clinical trial. So we started a clinical trial where we did show some activity of venetoclax just as a single agent. And then I think the very wise decision was made to then combine venetoclax with some drugs that we already knew worked, like decitabine or azacitidine or Ara-C. And then those became very successful clinical trials, and that eventually led to the FDA approval. So for me, I was able to see it all like 10 years beforehand when I was a postdoc. We were actually testing in our laboratory the great-great-great-grandmother of venetoclax, before when these drugs were just getting going. And it was I have to say, I'm an oncologist as well as a lab researcher, and it's very gratifying to see that you could do something in the laboratory and like kind of see every step of the process all the way through to FDA approval. So like that's something that I understand much better than I did before.